Good morning, and welcome to this second edition of pharma.be's virtual roundtables. Today's roundtable will cover a topic that you voted for in our last session in June. And that topic is the future of science. What's next? My name is Brigitte Nollet. I'm the General Manager of Roche Pharmaceuticals here in Belux. I'm also a member of the Board of Directors of pharma.be and the task force lead for the communications team, hence my role here today as your moderator. I'm also a patient suffering from psoriatic arthritis since my early 20s. And I share this with you openly and upfront because today we're going to talk about science, and we're going to talk about science in healthcare. And these discussions will rightfully lead us to really important areas like health data, molecular pathways, pandemic considerations, healthcare systems evolutions, and regulatory evolution. And that's where the discussion should lead us. Evidence in our discussion is really where decision-making is going. But I simply invite us, all of us as panelists, all of us as listeners, to remember that at the end of this debate, at the end of this discussion, we are really talking about how to support people, patients like me, in getting the new medicines and the new healthcare solutions they need to live healthy, to live happy, to live productive lives. This is about how science serves life. And my own experience tells me that new medicines, I know that with the new medicines, that's why I'm here with you today. I would not be an active and contributing member of society without the future of science, without advancements in science. So my invitation to all of us today is to consider that perspective in our dialogue and as we conclude. And that's why I'm so delighted to be with you and to be moderating today's roundtable with such a diverse audience. And we're also delighted to present to you today's top panel of experts who are going to bring their own unique point of view to how the future of science is shifting and how the future of healthcare can bring us together and what actions we need to take collectively to ensure that we're prepared for this future. So I just need to walk through a few logistical pieces before we turn it over to the panelists. The first is to remind you, of course, that this session is being recorded. The second is to let you know that at the end of the session, if you could just stay with us for 30 seconds longer at the end of the session, there is a survey that will be shared with you. And that feedback that you will give us is extremely meaningful in terms of helping us plan future activities. Each speaker will cover a specific element of today's topic for approximately 15 minutes, and then we will follow that with a panel discussion. So your questions are very welcome in the Q&A button that you will see at the bottom of the screen. We are really looking forward to your questions, and we are going to include as many as we can in the discussion. And if there's not sufficient time to answer all, we will make sure that we're able to reply to you individually after the event. So those are the logistics to share with you at this moment in time. Now let me introduce our panelists. The first person I'd like to introduce is Mrs. Geet Musch, who's the Director General at the Federal Agency for Medicines and Health Products. The second is Mr. Guy Oliver, who is Country President Belux and General Manager Oncology Belux at Novartis. And the third speaker, but our first speaker of this session, is Professor Muriel Moser, who is Research Director at l'Université Libre de Bruxelles. And if you allow me, Professor Moser, I just want to add a few more details to the introduction, and then I turn things over to you. Professor Moser studied biology, and in particular immunology, at l'Université Libre de Bruxelles. After a 3-year postdoctoral fellowship at the National Institutes of Health, that's the NIH in the United States, she returned to ULB and studied the role of dendritic cells in tolerance and the underlying molecular mechanisms. Professor Moser was Director of Research of the National Fund for Scientific Research and Dean of the Faculty of Sciences at ULB. She recently published the book entitled Vaccination, Biological Foundations and Societal Challenges. And I have to tell you, I can't wait to order the book and read it. So Professor Moser, thank you for being with us. The virtual floor is now yours.
Thank you very much, and thank you very much for your invitation. So we are now facing the third coronavirus-induced disease, which is COVID-19, and which is caused by the SARS-CoV-2 virus you see here with the chrome. This disease can be symptomatic and asymptomatic, which complicates the treatment, of course. And the outcome of the disease is very variable depending on the age and comorbidities, and with a higher lethality for aged people. And it caused quite a lot of death, with more than 4.8 million deaths in the world, which is much more than the two other coronavirus-induced disease that we have faced since 2000, which is the SARS-CoV in 2002, which causes 774 deaths, and the MERS-CoV in 2012, which caused 858 deaths. So the question is why? What are the characteristics of the SARS-CoV-2 that explain that pandemics, which was not the case for the other two viruses? And the next slide shows that normally people have shown that there was an inverse relationship between transmission, so the incidence of disease and the pathogenesis. And this is the case for influenza viruses and coronaviruses. So as you can see here, the transmissible viruses, like for example, the seasonal flu or the coronaviruses which are responsible for the colds, are very transmissible, but are not pathogenic. And it's the opposite for the MERS and SARS, so the previous two coronavirus diseases, which are highly pathogenic but not transmissible. Now, when you see the COVID-19 is in the middle, next slide, and it has been shown and published that the SARS-CoV-2, in fact, displays high transmissibility together with moderate pathogenesis. And this is exactly what we have seen for the H1N1 influenza disease, which has been which occurs in 1918, and it's called Spanish flu, and which caused between 20 and 100 million deaths. So that explains why the SARS-CoV-2 caused so many deaths. And it is, next slide shows that in fact, it's a matter on coronavirus tropism, because the report that has been recently published shows in fact that those cold coronaviruses and flu replicate mainly in the upper respiratory tract and cause a very mild symptoms. And so, whereas the SARS-CoV and the MERS-CoV, which are both pathogenic, replicate mainly in the lower respiratory tract and cause severe disease. And as you can see here, the SARS-CoV-2 can replicate in both sides, in the upper and in the lower respiratory tract, which probably explains the important consequences of this disease. So next slide shows that for the vaccine, it makes sense to choose as a target the spike protein, because the spike protein has been shown to elicit immune responses in, for example, in convalescent from the MERS-CoV on the SARS-CoV. It has been shown that an immune response was induced against the spike protein. The spike protein is expressed on the surface on the SARS-CoV-2 and is in fact the key for the entrance of the virus in our cells. I remind you that the viruses do not replicate outside the cells because they need the enzymes and the energy of our cells. So if The antibodies, for example, can block the spike protein. That means that it prevents the entry of the virus in the cells of the host. And there is also another mechanism, which is killing cells, so cytotoxic T cells, which can kill infected cells, which can therefore kill viruses which are inside cells already, because the antibodies cannot enter the cells. So there are two mechanisms of protection, two immune mechanisms of protection against the virus: antibodies that can act on extracellular viruses, and then killing cells that act on intracellular viruses. So the choice of the protein as a target made sense scientifically. And next slide shows that different technological platforms have been used to induce, to develop vaccines. So the first one is the messenger RNA, which, and this is important, is in fact modified. The messenger RNA is based on the discovery that modified RNA, messenger RNA, can induce a very good immune response in vivo and not too much inflammation, like has been seen for mRNA, which are not modified. And this is the case for CureVac, for example, who stopped the development of their mRNA vaccine. So the mRNA vaccines which are available, for example, in Belgium, are based on the modified mRNA technology, and this mRNA is encapsulated in lipid nanoparticles to permit the entrance in the cells. The technology has been developed by Katalin Carrico and Drew Weissman. The second technological platform is a viral vector, for example, the adenovirus vector, which is not replicating. And this is the technology used by AstraZeneca and Johnson & Johnson. And then there are also the proteins. Novavax has very good data about the phase 3 with the protein and adjuvant vaccine, and then other vaccines are based on the inactivated viruses, especially vaccines that are made in China. So next slide shows the first phase 3 data, which have been provided by Pfizer BioNTech. And those data very clearly show that the incidence of symptomatic disease was much lower in vaccinated people than in not vaccinated people. So that was the very first data, and that shows efficacy of 95%, which was much more than what was expected, because the OMS said that they would accept any vaccines with an efficiency of 50%. So the next slide shows the different data of the phase 3 trials for different vaccines. And again, you see here Pfizer and Moderna, the mRNA vaccine, and Johnson & Johnson and AstraZeneca with the adenovirus vaccines. And what is important to know is that first, the number of people or volunteers people for the phase 3, the number is much higher than what was required, 10 times more people than for the previous vaccines. So this is important to explain to hesitant people. And the main finding, in fact, is that the protection, again, hospitalization and severe disease is very high. It's close to 100% for all vaccines. And of course, it depends on the variant, it depends on where the phase 3 has been done. For example, some vaccines show a lower protection against variants in South Africa, for example. But in all cases, the protection against hospitalization and severe disease is very high. Next slide, please. So now we are in a Delta-dominated period, and for example, in Belgium, the Delta variant represents 100% of the sequence viruses, and it's highly transmissible. The viral load is about 10-fold higher, at least 10-fold higher than for the Alpha variant, which is the previous one, or the original virus, so which cause, of course, complicate things. So, but if we look at the data, the vaccine effectiveness is always very high. So, for example, this is for the Pfizer-BioNTech vaccine and the AstraZeneca vaccine. You can see that against all infection, the protection is still very high, 82%, 62%, and it's higher when we look at the self-reported symptoms. So for the symptomatic diseases, in that case, 86% and 70%. It's also important to stress the point that we now know that not vaccinated, but previously positive individuals are very well protected, even against the variant, this Delta variant. For example, to all infection, it's 73%, and to symptomatic disease, is 57 and 51%. So the next slide discuss about the host-to-host transmission. So we know, as I said, that the Delta variant is highly transmissible. And so what is the effect of the vaccination on the population? So the Cienzano data in Belgium from October 2021 showed that the risk of infection is diminished by 75% after vaccination. 75% is quite stable. And another important question is to test whether the vaccinated people still can transmit the disease. And so what has been shown in several reports is that the vaccinated individual with a breakthrough infection, so become infected, they are probably less infectious for different reasons. They are less infectious than unvaccinated individuals because the viral load is lower first, and also because the particles which they can emit if they are infected are probably less infection because they can be coated with the antibodies and therefore can be killed or can be, or the entrance to the other cells of the host can be diminished by the antibodies. But if we think about the type of vaccine, the vaccines which are used now are intramuscular SARS-CoV-2 vaccines. And those vaccines are optimized to mainly to reduce the disease, but not really to generate the mucosal immunity at the site of transmission, which is the nasopharynx. So because we know that the SARS-CoV-2 primarily spreads by respiratory droplets and aerosols, it would probably make sense to develop intranasal vaccine, mucosal vaccine. And mucosal vaccine would be very efficient to prevent the transmission because they would produce a different type of antibodies, which is a dimeric IgA, which very efficiently can inhibit the virus at the level of the mucosa. And in fact, so the future approach in transmission to block the transmission would be mucosal vaccine. And indeed, for the moment, there are several intranasal vaccines which are a development. And a very interesting paper published in Cell in 2020 has shown that the intranasal version of the AstraZeneca vaccine, so an adenoviral vaccine, can very efficiently prevent upper and lower respiratory tract infection, much more than the same intramuscular vaccine. So this is probably something that's going to be of great interest for the future. So the next slide asks the question of whether it is important to vaccinate adolescents and infants because we know that they are, of course, less at risk, that they have a very low risk of dying, or having severe symptoms. But the Delta variant, of course, complicates the things, and because the viral load is at least tenfold higher. And then one thing is that, and what I think is that it would be probably a good thing to push the vaccination even of young people, because they should prevent the risk of long COVID after infection. And the risk of long COVID is quite high, and it is quite high across all age. I will show you some data in the next slide. It's about 20%, but some people report 50%. So the reason why we should, I think, vaccinate those young people is because we want to prevent the long COVID, and also because they would, of course, participate to the collective immunity, which would reach about 80 to 90% of the population immunized if we want really to stop the replication and the transmission on the virus. So this slide shows that even in very young people, there are persistent symptoms after 6 months. So this is probably very specific to the COVID-19. And it's very important because even in the 16 to 30 years people, or 31 to 45 years people, so very young people, about 50% have some persistent symptoms after 6 months. And for example, among the symptoms, there are fever, but mainly palpitations, and then disturbed taste and smell, very high fatigue, and concentration problems, memory problems, and sleep problems, headache, and so forth. So this long COVID happens quite often, and I think it's a good argument to favor vaccination of the young people. So the next slide shows that we know that some hesitant people hesitate because they are really afraid of the adverse events caused by the vaccination. However, I'm always a little bit amazed because they do not really consider the adverse events after infection. And they are quite important, much more than the adverse events after vaccination. And that is shown here. So a report in the New England Journal of Medicine compares the adverse events after infection. And when they are higher after infection, they are in orange. And when they are higher after vaccination, they are in blue. And you can see that except for the lymphadenopathy here and the herpes zoster infection, the adverse events, and especially the severe ones, are much higher after infection than after vaccination. So of course, this is an argument to encourage people to be vaccinated. So the next slide. So it is quite clear that we're going to have other pandemics, either because of influenza viruses or because of the coronavirus, because 75% of new and emerging human infectious disease are zoonotic, which means they are zoonosis, which means that they are caused by germs that spread from the animals to people. And that the beta coronaviruses that circulate now in different animals are quite genetically similar to the SARS-CoV and the SARS-CoV-2. And they can use the same human receptor, which is the ACE2, as receptor to entry the cells. That means that they have a high potential to be transmitted to humans, so we have to be ready. So what we can do, of course, is create a vaccine for each coronavirus, but create a more universal coronavirus vaccine that would be a pan-coronavirus vaccine, and that would protect against different strains. And is that possible? Yes, we have some evidence for a proof of concept. For example, the first evidence has been come from those people which have are convalescent after the SARS-CoV, because reports have shown that they have cross-neutralizing antibodies. Those cross-neutralizing antibodies are able to neutralize so the SARS-CoV-2 and also other beta coronavirus isolated from humans. So that means that it's possible to develop a pan-coronavirus vaccine. And also another evidence comes from a report from macaque. So they show that immunization of macaque with nanoparticles conjugated with the binding domain from SARS-CoV-2 and adjuvant or from mRNA vaccines, those macaques after immunization are able to elicit cross-neutralizing antibodies. So this shows that this would be an interesting approach for the future. So the next slide shows that what should be done to be prepared for the future. So of course we have to survey the pathogen, to make data collection and to analyze how they can spread to humans, what receptor they recognize and how they can be blocked. And of course, vaccines and therapy have to be developed. For example, Anthony Fauci proposed to create prototype vaccines against about 20 families of viruses to speed up the development of vaccines in case of emergency. So if we think about the achievements in US as compared to European countries, so US has launched a warp speed operation, which was an operation that puts together private and public partners, and they had a great success in delivering approved vaccine in less than a year, because they were able to coordinate the efforts across the whole value chain, from basic research and then large-scale manufacturing, large-scale distribution, and also across the public and private sectors. And also they provide a high budget to do so. So if we look at the European countries, although we clearly have a more resilient social models, but we have lower levels of financial investment, that's very clear. And this morning I heard that the Belgian government has proposed to increase the budget for research, mainly concerning the viral infection, of course. And we also had not an optimal ability to ensure the coordination between national and European funding schemes. So that could be clearly improved. So the European Commission has recently announced the formation of an European Health Emergency Response Authority, which will, of course, help to manage and to coordinate all the efforts for the next pandemic. Of course, we have to improve the public health guidance and communication, and also addressing inequality also in the whole world, because for the moment, very few people are vaccinated in to be developed regions, which is of course not acceptable. So to finish, I would conclude that the vaccines are clearly very efficient, that they are very efficient to prevent mainly severe disease and hospitalization. And for example, the data provided by Cienzano in Belgium have shown that even now with the Delta variant, the breakthrough hospitalization remain very low, and the breakthrough infection are also quite low. But we have now really pressing unanswered question, and in particular, two of them. The first one being, what is the extent to which the emerging variants will evade eventually the existing immunity. And for the moment, all variants can be efficiently combated by the vaccine, so that's a good news. Of course, you have to be very attentive to that. And the second important question is whether the immunity will wane over time. And the data are still reassuring because we have quite few breakthrough hospitalization and breakthrough infection, but in some countries it has been shown that indeed the immunity wanes over time. This is why it is forecasted to again give a third dose to older people. So thank you very much for your attention.
Thank you. Thank you, Professor Moser. I don't think you can step away from your presentation without feeling your call to action. And what I really appreciated from your presentation was how you walked us through the past pandemics, the present data and what is taking place, and painting the future for us and what we need to be thinking about and looking for. And in particular, I appreciated from a futuristic point of view, which, as you've rightly said, it is not like we can say a post-COVID world, because we will be living with this long COVID. And I thought you were really clear on that. This is an issue for the future. And it's an issue that we, beyond the scientific side, health systems and healthcare will have to evolve to understand how long COVID will continue with us. But also your call, which was that different pandemics will continue, and we have to learn from this one and be very ready for the future pandemics to come. And I really appreciated your explanation on these different vaccines, the pan-corona vaccine or the mucosal vaccines, and what that could do. I think is really fascinating. And finally, the way you concluded, which is really a call for collaboration across the globe and across Europe in our case, which is we really need to be thinking about health data, how we collect this information, how we do it in a safe way, but in a way that allows us to answer some of these unanswered questions around the new variants and immunity over time, because it's that health data and it's the collaboration and how we join forces across public, private, and even the not-for-profit sector that we find that way to equalize across the globe, equalize across Europe, and collaborate for the future. So thank you very much for sharing those insights. I thought that was really, you were also so passionate about it. You can't help but feel really strong about where we are headed. So thank you for sharing that. I'm now going to turn it over to Guy Oliver, but if you allow me, Guy, just a few moments for your bio. Guy is a pharmacist, originally from New Zealand. He has over 15 years experience in the pharmaceutical industry, primarily in clinical development and medical affairs across multiple disease areas. He is passionate about science and pharmaceutical innovation and has experience across multiple and different therapeutic platforms. Currently, he is the general manager for Novartis Oncology and country president for Belgium and Luxembourg. And in this role, he is leading the late-stage development and commercialization for a broad portfolio of medicines. And beyond the science of innovation, Guy is also very passionate about evolving healthcare systems to ensure sustainable patient access of those innovations. And as a fellow board member with Guy, I can only attest to that passion, and really looking forward to your talk, Guy, about how our healthcare systems need to evolve to these new therapeutic platforms. So the floor is now yours.
Fantastic. Thank you very much, Brigitte, and thank you for the invitation. And also thank you to Professor Moser for your presentation. I think when we step back and realize that those COVID vaccines were manufactured, developed, and commercialized in under a year, it's absolutely amazing. And if we think about how we can apply those learnings to other therapeutic areas, I think the potential is huge there. So if we can just go to my first slide, please. I'm going to look at our sort of evolving science in the pharmaceutical industry from a very macro perspective. And I quite like this cartoon here because it kind of shows us how things have been evolving with regards to our pharmacological approaches to treating disease. And on the left-hand side, you can see small molecules. And small molecules over the last 70 years have absolutely revolutionized how we treat disease. Everything from infectious diseases to big population health challenges like cardiovascular disease and cancer, small molecules have had a massive impact on those diseases. So if we consider antibiotics, or we consider statins for high cholesterol, or tyrosine kinase inhibitors for cancer, there's multiple examples of targets that we've identified that are druggable, and small molecules that can target those targets. So I'm talking mostly around molecules that target transmembrane receptors or enzymes, and they're usually agonists or antagonists. So they improve a signaling or they stop a signaling. And it's been extremely effective. These medicines, however, are generally things that we have to take chronically. So we have to continually expose the disease to these medicines in order to have the therapeutic effect. They're also extremely convenient because they're usually orally taken. We move over a little bit, in the late 80s and early 90s, biologics became a lot more common as a therapeutic platform in drug development. Primarily monoclonal antibodies, and one of the biggest, actually the biggest drug in the world ever is a monoclonal antibody, a drug called adalimumab, HERA, which I forget how many indications it has, but I think it's something like 20 different indications across a huge range of autoimmune diseases. And medicines like that have enabled us to target new targets within the body. So things like inflammatory cytokines can now be controlled by monoclonal antibodies. However, like small molecules, they're usually taken chronically, sometimes with less frequency than a small molecule, but usually chronically throughout a disease. And what's really exciting is the rest of this cartoon, because we're now emerging into different therapeutic platforms. And as has already been described, we have new approaches. Gene therapy, which just 10 years ago seemed like science fiction, is now a reality for us. And there are now a number of gene therapies that are not just in the clinic, but are actually licensed and reimbursed in countries, including here in Belgium. And these gene therapies usually add an RNA molecule or some type of influence, so we can either silence or express our genes in a different way. And what's happening in early research and early clinical development now is gene editing. And this is when we really start to edit and control the genome. So things like CRISPR technologies, Cas9 technologies are in phase 1 and phase 2 clinical studies as we speak. And the potential of these platforms is massive across multiple diseases. And then finally, cell-based therapies. And this is also something that seemed science fiction only 10 years ago. But it's amazing that in Belgium today, we actually have 2 CAR-T therapies available for patients. And these are therapies where we take immune cells, in this case T cells, out of a cancer patient. We re-engineer those T cells, and then we put them back in the patient to fight the cancer. And the fact that this has not just been taken through the development stages, so the efficacy and safety has been proven, but more, the actual logistical challenges of these medicines have been overcome. Because the advantages of small molecules and biologics is that they're generally quite easy to make, they're quite easy to distribute around the world, and they're quite easy to administer to patients. The challenges we have on the right-hand side of this cartoon are that these medicines are difficult to make, expensive to make, take a long time to make, are usually quite complex to administer, and are definitely complex to distribute around the world. So it's an exciting time, and I really feel we're on the cusp of a tsunami of innovation, because the utility of these new platforms is very, very broad. If we go to the next slide, we can see that actually we now have the ability to interfere with diseases at different stages. So transcription, translation, and transduction, or even direct cell therapy. And I won't go into details on this, but we can definitely discuss it more on the Q&A, these individual approaches and some of the challenges around them. If you go to the next slide, please. The other topic I wanted to discuss today was really around our movement away from general conventional medicine to more precision medicine, and ultimately to personalized medicine. And this has been a huge change over the last few years. And I believe in the next 10 years, this will develop at an increasing speed. So first of all, our understanding of disease, our understanding of the pathophysiology of disease has increased greatly. We also have the ability with these new therapeutic platforms to target individual mutations and individual subtypes of disease like we haven't been able to before. So instead of treating all patients with a disease the same way and hoping that most of them will have some effect and some positive response, we can now identify different genotypic, phenotypic, and other disease characteristics to give us a good idea about what medicine should be used for that patient. So this generally increases the efficacy of the medicine, decreases the adverse events or decreases the tolerability issues that can sometimes be had. But we also have the ability to understand the prognosis of the disease better. So we have the ability to do this. One of the huge challenges we have is around collecting the data and so we can implement this in general practice. So Belgium is in a unique position because we have very, very good clinical, phenotypic, and genotypic data. The challenge that we have is actually combining this data into a big enough data set so we can use it in normal practice. But definitely the science is going there, and I think we need some efforts around the data in Belgium so we can harness this. And then my final slide is just to propose some of the questions that perhaps we can discuss in the Q&A. So when you look across all of these different therapeutic platforms, I've got them at the top here: small molecules, biologics, gene therapy, gene editing, cell therapy. And actually one I didn't discuss is radioligand therapy, which is a very different therapeutic platform, but one that also has huge potential. Radioligand therapy is where we use molecules to bring radiation directly to the target in cancer. And there's good data now that shows very high efficacy in some tumors, like neuroendocrine tumors, prostate tumors, and some others. So if we look across these different platforms, I believe that the science is accelerating, and that's fantastic. But actually, as Bridget said at the very start, getting these innovations to patients is our primary goal. Otherwise, they just sit on the shelf and they don't help humanity at all. So some of the challenges that we can see, first of all, as we go down to more precision medicine, as we identify additional targets that are druggable, we usually go down to smaller patient populations. So doing clinical trials can be more difficult because there's fewer and fewer patients. From a regulatory perspective and from a reimbursement perspective, understanding the true efficacy and tolerability of these medicines gets more challenging because we can't have big clinical programs with tens of thousands of patients. So we introduce some uncertainties about the efficacy and safety of the treatment. So we need to be more flexible in how we assess these medicines. We also need to make sure that we have a robust post-marketing authorization safety plan, so we can truly use real-world evidence to understand the efficacy and safety of these medicines when they're used in a larger patient population. When you think of biologics, biologics have revolutionized many, many diseases. And the exciting thing about pharmaceutical development is not just getting that innovation to the market, but also when the patent expires. And when the patent expires, we have the ability to share this innovation with a much larger population. And that's an important part of our pharmaceutical innovation in our pharmaceutical life cycle. With biologics, it makes it a bit more difficult because these medicines are more difficult to make than small molecules. So understanding the interchangeability and understanding how we can ensure that biologics are spread to all parts of the world is a challenge. And this challenge gets more and more difficult as you go along these therapeutic platforms. Imagine for a moment when CAR T therapies patents expire. How are we going to genericize and send CAR T therapy around the world? It's a very, very different challenge from just genericizing a small molecule drug. So there's some things we need to consider. Another big challenge we have is that as you move along these therapeutic platforms, they move away from being chronic treatments to being one-time treatments. And that's absolutely amazing for the patient. It's also amazing for our healthcare systems because these patients can sometimes be treated with one infusion and then live a normal life. The efficacy of these one-time treatments is usually very, very high. But of course, the budget impact can also be very significant. So if you think about gene therapies, think about cell therapies, they're usually very, very cost-effective medicines. However, the budget impact is really difficult. So instead of paying maybe 20,000 euros a year for a cancer treatment over 20 years, a healthcare system is now being asked to pay €1 million up front. However, it's only one-time treatment, so that cost is concentrated at the start of treatment. So we need new and innovative ways of approaching how we reimburse these medicines. The other things to consider are around center expertise and center setup. So these medicines, if you look at the far right-hand side of the pictures at the top, cell therapies, radioligand therapies, they need specialized centers to be able to administer these medicines. These are medicines that are not going to be able to be used in every hospital in Belgium. So we need to think, where are the right centers? How do patients get access to those centers? And how do we ensure that there is the right expertise to be able to administer and manage these patients? And one little example which can cause big problems is with radioligand therapy, you have radioactive waste that you need to get rid of once you've done the therapy. So what hospitals have the ability to be able to manage something like that? So we really need to move away from our traditional way of looking at treatments, think about more specialized centers who have the expertise and the logistics to be able to use these new therapeutic platforms. And the final question we have a lot, particularly in cancer, but also in other diseases, is with all of these innovations coming through, which is fantastic for patients, fantastic for the healthcare system, how do we use them together? So do we start on the left-hand side and move to the right-hand side because that's historically how we discovered these platforms? Do we combine different platforms? Which platform should we use first? Which platform should we reserve for patients who maybe are resistant or refractory to previous platforms? They're questions that we have and questions we really need to answer through robust clinical trials, but also really robust real-world evidence as these new platforms get used in Belgium. So in summary, I believe we're at an extremely exciting time on the cusp of these amazing innovations, not just coming into clinical trials, but actually coming into real-world settings in Belgium. There's a number of challenges we need to face in the healthcare system to be able to really maximize the impact of these new platforms for patients. And I'm excited to see how we meet those challenges. So thank you, Bridget.
Thank you, Guy, for that inspiring talk. And I fully agree with you. It is a wonderful time to be in the health sciences right now, given the exciting research that we have, the exciting innovations that are coming forward. And I really appreciate the way you walked us through small molecules to the more complex ones and what they mean in lay terms for people and how we move from chronic to one-time therapies. And I appreciate as well your comments about how that means we're also getting more and more personalized, which is a wonderful thing as a person living with a disease to see that personalization, but it doesn't come without some complexity, which I think you very fairly identified. And I think, you identified challenges which are also opportunities. And I think the call to action actually from both speakers so far has been, how do we collect health data to be able to guide decision-making within these challenges and opportunities? And how do we collaborate together? Because there's no one part of the healthcare system that can solve that list that you showed us at the end by themselves. So how do we do this together? And the willingness of everyone to come together. So thank you for that, Guy. I really appreciate it. And it's a wonderful segue to our final speaker, Mrs. Mush, in terms of how do we continue to evolve our healthcare systems and think about the perspective of the health authorities within this future science. So allow me just a moment to introduce you, Mrs. Mush. Mrs. Mush is an industrial pharmacist and obtained a PhD in pharmaceutical and biomedical sciences at the Free University of Brussels, le VUB. She joined the pharmaceutical industry for 8 years, where she was responsible for the chemical and pharmaceutical analytical activities related to the development of new innovative medicines. She then moved to the Federal Public Health Services as a senior quality assessor and assisted in several projects related to AMA and the CHMP. In August 2004, she became responsible for the R&D department within the Federal Agency of Medicines and Health Products in Belgium. And as of February 2009, she was designated as Director General for the DG pre-authorization. She is involved in different working groups related to unmet medical need, early access, and clinical research in a broad sense. And she is also actively involved in the pharmaceutical strategy in the European Medicines Regulatory Network. So thank you for being with us today, Mrs. Mush, and the floor is now yours.
Okay, thank you very much, Brigitte, for the kind introduction and a warm welcome to everyone in the audience. So, and thanks to the previous speaker who already prepared the scene on challenges where we as authorities are faced with. And my angle is more coming from where are we at regulatory authorities, licensing agencies. That's not the reimbursement aspect. I will not deep dive on that, but more the licensing agencies. And I'm happy to walk you through. So if I have the next slide, then we will start a bit. We have a very ambitious European Commission today, who is very reactive, of course, on the COVID-19 pandemic. And you will find a lot of points where we can liaise on what Professor Moser told us and also what Mr. Oliver told us. So as you know, probably due to the COVID-19, there is a proposal to make a regulation on reinforce the role of the European Medicines Agency in crisis preparedness. As already said, there is a HERA, so they are really working on European Health Emergency Preparedness and Response Authority. There is a lot of things to do on clinical trials, and an initiative is prepared at European level to make a clinical trial transformation initiative. The Commission is revisiting the pharmaceutical strategy because, of course, we should, as regulators, move to a more agile and flexible system in legislation. Blood tissues and cells regulation is revisited. We have the European cooperation on health technology assessment, which is progressing very well. We have a huge project on health data. So you heard from the previous speakers the significant importance of also building on real-world data and real-world evidence and combining this with the classical clinical trial data. So that's a huge project as well. COVID-19 therapeutics is now one of the points also. So we expect a list of 10 promising COVID-19 therapeutics issued end of this month. And I will share also with you a few initiatives, but more at the level of DG Research and Innovation. Next slide, please. So this gives you a flavor of the mandate of the European Medicines Agency. So in emergency crisis situation, they will work on, as they did already a bit now, but now their mandate is enlarged. So they will be able to really monitor and mitigate the shortages for medicinal products, but also the critical medical devices. Ensure timely development and really speed up the authorization of potential candidates. And I will come back on that in the next slides, because we used already some systems during the COVID-19 for that. And they will also provide the structure for the expert panels to assess high-risk medical devices. Next slide. So this is in fact the HERA Incubator who has been launched. And in the next slide, we can see on which 5 pillars, in fact, they will work. So the one is the rapid detection of the new variants. The second one is a swift adaptation of vaccines. A lot has to do work will be necessary to really set up the European clinical trials network, really to speed up setup of those trials in Europe in a more structured and guided way. And there will be the launch of the Accelerate, where Belgium is participating. So I'm very happy for that. And of course, the fast track of the regulatory vaccine approval process. So you had already an oversight of the different vaccines, which was presented by Professor Moser. And it's true that we really had to work on a new system. So the rolling review system, which was handled by the emergency task force, really they did really a good job in facilitating the fast authorizations of these new vaccines. Ensuring of also support to manufacturers, because that was a difficult point also during the pandemic, that okay, you can develop and you can foresee some manufacturing sites, but the capacity and the flexibility remains still a challenge for the pharmaceutical industry. And there we come, upscaling of vaccine production and swift delivery. So they will really build a pillar on facilitate tech transfer, provide capacity support. And also where we worked also was on the APAS, what we call the, how should I say, the budget agreement, the advanced purchase agreement, where even with few data, we had to say, okay, we will already foresee to buy for our member state several types of vaccines. This system will be optimized as well. Next slide, please. Here, this gives you a view. HERA's task will, of course, not only work in an emergency phase, but also in a preparedness phase, and a bit more in detail what they will do. But of course, and they will be hosted probably in the European Commission. So this is really speeding up to set up this HERA very soon. Next slide. Clinical trial regulation, as I said, it's really an important cornerstone for Europe, also for Belgium, but certainly also for Europe. You know that we will go live end of January next year with new regulation. There is an implementing act on safety, which will be adopted as well. There are 2 joint actions where Belgium is coordinating the expedited assessment of trials of COVID-19 therapeutics, and there will be a second one in the second wave for the coordination of safety assessment. And as I said, there will be an initiative taken by the Commission together with EMA and the national competent authorities to go ahead with clinical trial transformation initiative to really make Europe stronger in this environment of clinical trials. Next slide. Yes, and then as I said, we have, in fact, a pharmaceutical legislation which is rather conservative, and the Commission did in fact a survey really to detect the different issues which should be tackled to make it more flexible and more, how should I say, agile. If we see all those innovation coming to regulators, this really needs a reflection. And in the next slide, you will see they in fact selected 12 to 13 topics where they will work on. And if you look, the centralized procedure, which is in fact the way to go for innovative authorizations, is revisited. They will look also specifically on new manufacturing methods, also on real world evidence, and all those issues will be optimized, and they will also reflect on a new way of working. So before making a huge regulation, they will rather work with sandboxes, so to try out some legislation and to make it more flexible and more in an iterative and flexible way. In the next slide, I go now from G-Santé to DG Research and Innovation, and I think there it's really good to see which, if I listen to Professor Moser, it's perhaps true that the funds are not in the same way structured or organized as in US, but anyway, I think we see some good initiatives, and we have the European public-private partnership between Europe and the health industries. For instance, pharmaceutical companies, including the vaccine sector, but also the medical imaging, radiotherapy. What Mr. Oliver said, the medicinal product, it's a small part. We should really see it in a more holistic view. And you see here some initiatives which reach out to these sectors. We have the medical technology companies, which of course are very, very important, and the biotech coming a bit to the biologicals. So this is good news. And in the next slides, you see some other initiatives. So they also have a budget of more than 100 million euros specific for the coronavirus research and innovation project. So these calls are launched now from HERA. The important really to make better structured clinical trial network via Vaccination, via response and recover. We learned that during the COVID that we really have to work on that to really speed up those trials and to be able to gain as fast as possible the correct data. The wide adaptive platform trials, so the complex clinical trials, is one of the spearheads of this clinical trial transformation initiative. We really work on that. And then the chapter on real world data to complement regulatory decision making, which is really for regulators a new kind of expertise we need to build, and we are aware of that. But that's really one of the main challenges for the European licensing agencies. Okay, if we move to the next slide, I think there we have indeed, I share with you a bit the regulatory strategy which has been elaborated by the European Medicines Agency. And you see 5 important strategic goals. And if we take the 2 from above, on the left-hand side, you see how will we catalyze this integration of science and technology, which is really at a high level speed, how will we insert that and catalyze that on one hand, and how we will enable as regulators research and innovation in regulatory science. And if we move to the next slide, you can see it's in fact a view which is elaborated by the European Medicines Regulatory Network. In fact, they say, okay, we should look at 3 different angles. The first one has to do with the disruptive medicinal product development, production, and supply. The other one is digitally enhanced evolution for evidence. And the last one is in fact the coherence of the scientific advice, downstream assessment decision making. So really making the link with the players and the health technology assessment bodies as soon as we can. And in the next slide, you will see what do we mean by this disruptive medicinal product. Mr. Oliver already touched upon several aspects on that. First of all, we go away from this idea to have an active substance in a very, very small definition. And you can see, okay, what are the evolutions where we as regulators are confronted with. For instance, immunopotentiation agents, the adjuvants, permeability enhancers, more and more the complex drug delivery systems where we have to see how we assess those applications. If we go to the next slide, the next slide, yes. More the novel manufacturing and supply methods. What are the new innovations coming on? We see platform technologies for vaccine, for advanced therapies, decentralized bedside manufacturing, the use of artificial intelligence, even in manufacturing, nanotech, additive manufacturing, 3D printing, and so on. So you see a lot of issues coming on this side too. And finally, if we look to combination products, there you really see the challenges, and we really should touch and reach out to devices, to in vitro diagnostics, to all other domains covering these complex drug delivery systems. And that is really a challenge, and that's the reason why in the mandate of, for instance, the European Medicines Agency, the link is made to these new technologies to be able to be agile enough and in time enough to make it happen in a regulatory pathway which is correct. Okay, the second part is more, okay, how will we build up our evidence? So there we have, in fact, three major cornerstones. One is foster the innovation in clinical trials. That's why the clinical trial transformation initiative is there. We have the enabling of all the digital clinical data generation. And as you know, there are a lot of projects. You have the Darwin project, you have the big data project, which is really launched and where we are working on, and we have to promote this real world evidence. How do we take this data in our decision making? Okay, next slide is more the really importance of having a coherence of scientific advice, but taking already the downstream assessment partners in mind. And there we should, that you can see we have to contribute to HTA's preparedness in a very, very early stage. We should integrate the scientific advice during the life cycle of a drug, and that's a bit what's also alluded to by the previous speaker, to say, okay, the life cycle of having one time in a regulatory pathway where you have your marketing authorization application is not, it's gone. We should work more in an iterative way and really learn from those data to see it's correct, learn from it, to put it in new R&Ds, and that's really another way of looking at it. And then, of course, we have everything which has to go with the borderline product. So it's a bit, how should I say, segregated in the European regulatory framework, and we should really try to make it more harmonized. Okay, I think that's it for this part. And then just as an illustration, I will not spend too much on it in sake of time, but just to show that, in fact, in Europe, we have what we call a regulatory network of agencies, EMA together with the national competent authorities, but just to show that, in fact, the strategy from those national competent authorities under the umbrella of the heads of medicines agency is fully aligned with the strategy we just talked, and they, for instance, have a specific topic on innovation, but also an important one on the sustainability of the network, because we as authorities must be able to really use our budget and resources in the best way. So that are really two important objectives for us in this regulatory framework. Okay, and then finally, I go to, you can go to the next slide, please. What do we do as a national agency? Just a bit with the relation with innovation. We have at our agency a national innovation office, which has the aim of being a key facilitator or really accelerator to really facilitate the access to innovative drug and healthcare products to patients. We also try to work that Belgium in Europe remains an attractive ecosystem for the R&D, and we identify together with the colleagues in Europe the emerging scientific and technological trends. So that's really important for innovators to know that the first point of contact for help or coaching to the regulatory framework can be done by this National Innovation Office. And we are, of course, one of the colleagues working in a European innovation network, which makes it more efficient and worthwhile to work together on this issue. Okay, and next slide, I think, gives you just a bit an idea because previous speakers focused on viral and oncology, just to say, okay, we see this also in our work, in our daily work. We have more than 50% of our requests for advices are in those two domains. Of course, the COVID-19 has, of course, played an important role in 2020. So we received a lot of applications on that. And next slide gives you an idea that 38% of our applications were related to vaccine 19, which really illustrates that Belgium is active in this field, and we had 10% onco-related advices, mostly on the use of complex trial designs. And finally, I think the last slide gives you a bit an idea. We are also, of course, working at the European Medicines Agency at the Scientific Advice Working Party. So we do more than 20% of the advices there. We have really a very good team. And you see that most applications are in oncology, but also in vaccines. And that brings me to the end of my presentation. So I hope I give you a big view of the evolutions at Commission level, at EMA, and national level, and that you have an idea where we as regulators have to work on. So,
yeah, absolutely, you did. Thank you very much, Mrs. Moosh. And what I'm really struck with is, first of all, I think we have to thank you and all of the regulators and all of the health agencies across the world, and for us across Europe and here in Belgium, because you mentioned this agility and this flexibility. And I think we learned so much in the last 22 months about how to think differently with our processes. And you were all at the front line trying to help work with the different scientific partners in the industry to ensure that the science was understood. And you mentioned the words in an iterative way. And how do we keep that going? And what do we learn from it? And how do we keep that moving? And I really appreciated your transparency with the 3 pillars around disruptive medicine, digital enhancement for evidence, and that downstream assessment. How do we keep learning and partnering? So again, that theme of collaboration and evidence coming through in your presentation as well. So this takes us, dear listeners, to some of the questions and answers that you have submitted, and please continue to submit them. We have a few minutes still for questions. And one of them, perhaps, Professor Moser, comes to you back to the vaccine piece, which is certainly, you spoke of 12 and up and you spoke of vaccinated and non-vaccinated, and the question has to do with children. And whether you would be willing to answer the question, does the same apply for children that perhaps, what does it say here? What would you say the same apply for children, that they probably would be meaningfully less infectious if they were vaccinated versus not vaccinated? The same piece you were saying earlier. How does it apply to children?
Yes, so I'm not pediatrician, so we should ask, of course, to the doctors, but at an immunological point of view, I think that it's a good idea now to vaccinate children from 12 years old. And if you look at the numbers, it's amazing, in fact. In Belgium, the 12 to 15 years old are vaccinated at 65%, which is amazing. In fact, it's very, very high. And already 69 have received 1 dose, so 65 2 doses. If you look at the 16 and 17 years old, it's 74% are fully vaccinated. So maybe for the moment, my feeling would be that I would vaccinate over 12, see what happens in other countries for younger people. One doctor said to me that maybe Novavax, which is the protein with adjuvant, would be better for the younger because it's very efficient, but with lower side effects. That's a possibility. So I think we need a clear advice from pediatrician, but that the fact that already now people over 12 are vaccinated at a high percentage, because it's more than 65%, I think should be maybe enough. And now let's look what happens to the different clinical trials, the different phases, for example, in the United States, and see what we do with the younger than 12. That would be a personal advice, of course.
Thank you very much. Yeah, definitely more to come, I think, on this specific piece, but thank you for that. Mrs. Mish, maybe we come to you. There's a question that's been submitted. What are the key areas for the Clinical Trials Transformation Initiative?
Thank you for the question. I think it's still work in progress, but on one hand, I think we need to be more efficient and harmonized in assessing already multinational clinical trials. That's one of the lessons learned, I think, also during the pandemic crisis. So that's for sure one. And also we should, and we learned that also in the pandemic, we should be more integrated, although it's still a national decision, we should be more integrated in the European system, you see, to make the link with the early scientific advices, the innovative task force, and really smooth that pathway. And we will have to invest a lot in new domains as clinical trial methodology, etc. Looking at the complex clinical trial, looking at decentralized trials, all those new initiatives should be handled, but and also making the trial, once we have an harmonized view where the regulation will for sure help a lot, but also to enable that those trials can be rolled out at the sites in Europe as efficacious as possible, and that we learned also was not always easy during the pandemic neither. So that's a bit in a nutshell some element, but it's still in elaboration, and we hope that we will have a final view end this year. Yeah.
That's great. Thank you very much. And maybe there's a question that's just come in, and I ask you again, Mrs. Misha, maybe yourself, Guy, to reflect on this question. It's about the value of innovation and how do we work together to ensure that we're offering access to medicines. And so maybe the question for you, Mrs. Misha, the angle is, you've talked a lot about what we've learned through COVID and how it was for this emergency timeframe, but can we take those learnings into the future and look at all access to medicines in this iterative, agile way? And then for you, Guy, what is it that we need to think about as industry and as partners around the table to be able to also ensure that there's access?
I will start perhaps, but not so. Yeah, I mentioned already some initiatives, but what we learned in the COVID-19 is different in my view than how we should see it in a regular world. I will explain. So in the COVID-19, we worked together, everyone, to really fight the pandemic and to really learn the disease and get insights. So that's good. But on access to patients, of course, you have the trials, which are already given access to patients and to be able to gain the data as quick as we can. So that's for sure one. But the other thing, which is different on the classical way of working, is a bit what I said, the joint procurements or the advanced purchase agreement, because there, from public health perspective, with limited data, and even when you will not have already a conditional marketing authorization, you have to decide as member states, but it has been also discussed between member states at European level, for which vaccine or for which antiviral we will invest. Yeah? That's another perspective in my view. Of course, we learned a lot on COVID-19, and everyone is making lessons learned, and we will learn from that. But I don't think that you can copy that, in my view, to the general way of okay. There, I think the more classical way, what it's already envisaged is really to have those health technology assessors in the network of the licensing agencies as a very early stage. But that's, and I hope you net know that co-work, between the HTA bodies will enable also to have some of them more and more present in the early stage of licensing. And then you have, of course, the other issues such as, okay, as is said, it can be that you have one application of your drug for a lifetime. How will you build reimbursement issues on that? But I think that Mr. Oliver can talk more about that kind of difficulty.
Yeah, thank you. So I think when we think about bringing innovation to Belgian patients, I first of all think about clinical trials. And I think we're blessed already in Belgium to have an environment that is attractive for multinationals like my company to place clinical trials in Belgium, and therefore to give access, very early access, to Belgian patients. But I really think we can't rest on our laurels. So I appreciated the comments from Mrs. Mush, particularly when we think about these new platforms. So ATMPs, one-time treatments, those sort of things. We need to make sure that our regulations allow for quick setup of clinical studies and to make sure that we can recruit patients quickly, because we are in competition with other countries when it comes to our clinical trials. And believe me, every other country in Europe and beyond also wants to be a pharma valley. So we need to remember that. And I know Bridget and myself, we see that all the time working for a multinational. So I think we need to definitely work on our clinical trial regulations to maintain our leadership place. When I think of actual reimbursement, as Mrs. Mush said as well, we need to get away from this view of we assess a medicine to decide its efficacy and safety, then we put it on the market. We have to take much more of a rolling view, that we look at something when we see that there's preliminary positive data, we license it, we reimburse it, but we don't just stop there. We continue to evaluate the efficacy and the safety, and we adjust as we go. So I think that's really important for these newer therapies. And then logistically, when it comes to the cost of these medicines, we realize that the one-time treatments are a big investment. So we need a more flexible payment approach and procurement approach. So some of the things we've seen in other countries are, instead of paying one time up front for a gene therapy or a cell therapy, actually paying over a period of time. So almost like a lifetime payment that you pay every year. Or even looking at more innovative ways of looking at outcomes-based payments. So if the treatment does not have the effect that we hope it has, that the payment isn't needed. So actually the medicine becomes free for the healthcare system. So there's a number of innovative ways we can look at reimbursement, but definitely with these new innovative therapeutic platforms, we have to look at them differently than traditional small molecule biological platforms.
Yeah, thank you very much. Thank you very much to all of the panelists. We have one outstanding question we'll make sure we answer directly with that person, and we'll follow up with you, Professor Moser, for that Q&A. But I just want to take the opportunity to thank all of the panelists. I think what you've done for us is you've helped us really understand and get excited about where science is headed in healthcare and how we can collaborate together to move forward. You've painted a picture of how strong Belgium is in terms of its contributions to healthcare, from the pandemic through to our clinical trial setting, through to how we work in a European network to be able to ensure that there's patient access. You've certainly highlighted for us that evolution from small molecules to larger one, and how the future is about personalized medicines, and how it's about how we support the right person, the right medicine at the right time, which I think is going to be really important for us as we go forward. And you've been wonderfully transparent in your discussion of the challenges and the opportunities that lay ahead. So the science is there, and I think you said it a few times, Guy, it's not so far off as we think it is. And how do we ensure that agility, flexibility that Mrs. Musch highlighted, to work together to bring it forward? So thank you for always keeping that patient in the center of our conversation, and that essence of collaboration, I think, was a really important thing for us to take away and hear. So with that, I'm just going to remind our listeners that there is the poll survey that's come up on the screen. We really appreciate your feedback. And to our three panelists, thank you again for sharing your expertise. And to everyone, please, I wish you a very happy, healthy, and restful weekend ahead.